Hello my dear students and welcome back to Excellence Batch, and I am your Diksha ma'am. So in the previous lecture, we have completed the entire immunity part. So now it's time to study some dreadful diseases. We have two diseases in our NCERT. One is AIDS, second is cancer. So let's get started with the first one, which is AIDS. So today we'll be discussing, as I've told you, AIDS. Alright, so first of all, what is the full form of AIDS? Because it's capital letters, right? So it's a short form. So the full form is acquired immunodeficiency syndrome. So its full form explains everything about AIDS. How? Let me tell you. First, acquired. Acquired means you take it from another source. Take it from outside. You are not born with this disease. It's not a genetic disease. Rather, you get this from another person who is infected. That's acquired. Acquired means you get it. Just like acquired immunity. Immunodeficiency, because here immune system is attacked. Immune system is attacked. Just like in every disease, some organs are attacked, like in heart attack, the heart is attacked, right? In hepatitis, the liver is inflamed. Just like that, in AIDS, basically immune system is attacked. That leads to a group of symptoms and disorder. You call it as syndrome. So syndrome means group of infections. So you will not get one symptom. You will not get one symptom, you will get number of symptoms. You will get number of symptoms in this disease, right? For example, the person may get tuberculosis, pneumonia at the same time, or the cancer, tuberculosis and some other infections, fine. So the first case of the AIDS was seen in US in 1981 in the homosexuals, right? So the first case, where was it seen? In 1981 in USA in homosexual males. Now, what is the causative agent of the disease? Is there any virus? Is there any bacteria? Who is causing this disease? So, what is the causative agent? Causative agent is a virus, a very deadly virus. The name is HIV. You must have heard of it. The full form of HIV is human immunodeficiency virus. What's the full form? Human immunodeficiency virus. So this virus was not the virus of humans from early stages. This virus was seen in monkeys. From monkeys, after mutation, it entered into the human. So from where we got this virus? We got this from monkeys after mutation, right? Now, if I talk about this virus, this is a retrovirus. This kind of virus is a retrovirus. What are retroviruses? Retroviruses are the viruses which have RNA as their genome. They have RNA as their genome. Now, what kind of RNA? Is it single stranded or it is double stranded? So, it is single stranded RNA. But how many strands? There are 2 strands. Now you will say, ma'am, then it is double stranded. No, double stranded RNA will be like this, something like this. 2 strands and they are attached with the hydrogen bonds. But single stranded will be like 2 strands, they are separate, they are not attached to each other. So this is double stranded RNA, this is single stranded. So this is how it looks like. So I am rubbing this one, fine. Another point to note here is that this virus is the enveloped virus. You will see a coating of protein on it. It's the enveloped virus. Now, how does this virus get into our body? Before drawing the structure of the virus in detail, let's see how this virus enters into our body. So what's the mode of transmission of this disease? What's the mode of transmission? That means how this disease gets spread. First of all, this disease gets spread by unprotected sexual intercourse with the infected person. If there are two people, one person is infected, another is normal, and they undergo sexual intercourse but unprotected, that means they are not using condoms, right? At that time, the body fluid of the infected person contains the virus. Now, body fluid during sexual intercourse gets into the another person will get the HIV, right? So, the first is through unprotected sexual intercourse. Through unprotected sexual intercourse. Second method, how it gets transmitted? By infected needles. For example, one person is infected with HIV and a doctor used needle for taking out the blood or giving any injection. If doctor accidentally or somehow uses the same needle to give injection to the non-infected person, the non-infected person will get HIV, because in that needle the blood has been in contact, right? And from blood the HIV gets stick to the needle, and through that it will go into the normal person. So through infected needle. And this is also seen in drug addicts. This is also seen in drug addicts, because drug addicts uses the same needle, right? There is a group of people taking the drugs through needle. So you don't know which person is having HIV, right? So by that it can infect from one person to another. Third is through blood transfusion. Through blood transfusion. So if a person is infected, so that person will be having HIV in its blood, right? So if that person give blood to any other person, the second person will get the HIV. So there was a case that happened in India. There was a female, she was pregnant, and she somehow got anemic during pregnancy, and doctor asked the female to have some blood, right? So the blood was donated by a very near relative, but that relative, whosoever he was, he didn't know that he is HIV positive, because you never get to know in 1 or 2 days, right? So that person was HIV positive. So that person's blood, when got to that pregnant female, she got infected, even the baby got infected, right? So always there is one more thing, that the blood should also be checked before transfusion. Third, from mother to fetus. As I've told you, the mother and the baby both got infected, because the HIV can pass through placenta. So from mother to fetus through placenta. If mother is infected, the baby can also get the HIV. So these are the various means by which the HIV can enter inside a person's body. Fine. Now, how does this virus looks like? Let's do some drawing. Okay. Since this is the enveloped virus, so it will have the coating of the proteins and glycoproteins. Yes, let me draw this. Okay, so this is the envelope, and this envelope consists of protein and some very special glycoprotein. So for example, there are certain spike protein like this. If you have seen the structure of the coronavirus, you must have seen in the movies or the media, especially, not in the movies, in the media they show the virus animation, and it's a small ball like structure which have like pins on it. So they are spikes. So most of the viruses, they have spikes on it, right? So just like that, it also have those. Usually those viruses which have such kind of the spikes, they are very deadly. These spikes are made up of glycoproteins. What are they made up of? Glycoproteins. Glycoprotein means protein conjugated with the carbohydrate. Protein conjugated with the carbohydrate. Let me draw this for you. Alright. Okay. So the protein that you see embedded inside, this is GP41 and this one is GP120. GP stands for glycoprotein. So GP stands for glycoprotein, and whatever number is there, this is their molecular weight in Daltons. This tells about molecular weight in Daltons. So you will see a lot of protein with such kind of name. If I am saying it is GP41, it has around 41 kilodalton of the molecular weight. Fine. Now, inside of that you will again see some protein coat. So that's a completely enveloped virus. So this is P17 coat or the protein 17 coat. Again 17 signifies its molecular weight. So this is the another coat. If you move inside of it, it's P17. If you will see from outside, this is the cut section of it. From outside it will look like it's a ball which have a lot of pins on it. Now you are cutting it from inside and seeing what it's inside, right? And then again we have another coat, and that's a coat of P24. So see, it's highly enveloped virus. It's P24. Okay, so that's P24. P stands for protein. GP stands for glycoprotein. Now, what's the role of this protein? I'll let you know when we'll study how it causes infection. Okay. Now, inside of this is present two strands of single-stranded RNA conjugated with the enzyme. Right. So, let me write this for you. This is single-stranded RNA. This is also single-stranded RNA. Fine. Now, what are these green ones? These green ones are proteins or enzymes, because enzymes are made up of protein. The enzymes, there are two kind of enzymes here, like proteases, and the most important, reverse transcriptase. As you all know, because you must have read it in the botany, in the viruses, retroviruses multiply by the technique known as reverse transcription, right? Normally DNA replicate, you call it a DNA replication, but sometimes RNA forms DNA. You call that technique as reverse transcription. So for that we need the enzyme, which is reverse transcriptase. Now this, this virus, how does it enters inside your body? How does it enter inside your body? You all know that now. It enters through these method. By any of these method, it enters inside your blood. Now, it will go to its target cell. Where it will go? It will go to its target cell. Now, the target cells are the cells of immune system. So, now what are the target cells of HIV? There are two cells in which it can invade, and these cells, they are CD4 cell. If you remember, I have told you CD4, this is a protein present on the surface of cell. One such cell is T helper cell. T helper cell, right? And second CD4 cell that you have in your body is a macrophage. And macrophage is known as HIV factories. Why? I will let you know. So these are the cells which are the target of the HIV. So how does it will enter inside these cells? Imagine this is your macrophage. This is your macrophage, right? Macrophage on its surface have a receptor known as CCR5 receptor. What receptor is this? CCR5 receptor. So if you don't know what are receptor, these are nothing. These are certain proteins present in the plasma membrane. What are these? These are just, we have the intrinsic protein, tunnel protein. So it's a kind of a tunnel protein. Okay. So receptors are nothing but these are made up of protein. So this receptor is CCR5. Now this is a macrophage. How the virus will enter inside macrophage? Virus is moving inside your body, and on its surface it has these spike proteins, and the outermost protein or a glycoprotein is GP120. is GP120. Now as you can see here, the GP120 and CCR5 will interact with each other. As a result, what will happen? As a result, the virus will send its genome through this receptor, right? First, they will do the recognition. What is this? This is GP120. GP120 and CCR5 of macrophage. GP120 of virus and CCR5 receptor of macrophage will interact with each other. During this interaction, the endocytosis will occur. Endocytosis of what? The RNA. Endocytosis means entry of something. So, RNA is present like this. So, through this RNA will enter inside the cell, and you call this technique as receptor mediated, listen to me very carefully, receptor mediated endocytosis. Endocytosis means that something is entering inside cell. Endo means inside, cyto means cell. Receptor mediated, with the help of receptor something is going inside the cell. What is going inside the cell? The single stranded RNA. The complete virus never enters the cell, only the genome enters inside the cell. So, through receptor mediated endocytosis, genome enters inside the cell. So, genome also contains enzyme conjugated with it. So, enzyme and genome both will go inside the cell. The entire envelope will move outside. It will not enter inside the cell of the human. Right. Now, I told you macrophage, they act as HIV factories, but T helper cell they don't. Why? Because viruses multiplies in macrophage. Viruses multiply in macrophage, whereas they destroy T helper cell. So, remember this thing, viruses destroys T helper cell and divide in macrophage. That's why they are factories. That's why they are factories. Fine, simple, no? You must have heard of lysogenic and lytic cycle of viruses. If you don't remember, go back 11th class book. There is a cycle that how does viruses divide? Viruses divide by two cycles. In lysogenic cycle, they never break the host cell. In lytic cycle, they break the cell. So, lytic cycle moves in T helper cell and lysogenic cycle moves in the macrophage. Okay, so let's move back to the story. Through receptor mediated endocytosis, the genome enters inside the cell of the human. What happened next? Okay. So, you can see here there is a retrovirus, there is a protein coat, virus is getting, virus is infecting the human cell, but it has only sent its RNA, it has only sent its RNA. Now, from RNA it will produce a DNA. So, this is RNA, from RNA what it will produce? DNA. One thing that NCERT has not mentioned is that there are two strands of RNA, both the strands separately will form two DNA. So, this is RNA, both undergo reverse transcription with the help of reverse transcriptase enzyme. You know what is reverse transcription? Formation of DNA from RNA. Formation of DNA from RNA. Reverse transcription will take place, DNA will be formed. The most interesting part is that both these DNA, they are complementary and they will join and form double-stranded DNA. Now, why there is a need of double-stranded DNA? Because this DNA has to get integrated into your nucleus, into your macrophage nucleus. This is your macrophage. Okay, this DNA has to go inside your nucleus and your nucleus has DNA which is double-stranded. So, for getting into the double-stranded, you also should be like double-stranded. Okay, if you want to go into a school, everyone is having one uniform, you should also have that uniform. So, for that double-stranded DNA is formed. Now, this double-stranded DNA, it enters nucleus. Okay, so virus send RNA, RNA form DNA, DNA become double-stranded. Now, it will add or incorporates into the host genome. Now, it has hijacked the host genome. After it has hijacked the host genome, what will happen? It will again undergo replication and transcription. What will happen here? Transcription. When the transcription takes place, again RNA will be formed. RNA will form protein and protein will form new viruses. Because what virus is made up of? Protein. GP120, GP41, 17, 12 and so, right. So, what this will do? It will form RNA, two types of RNA. So, one RNA will become genome and one RNA will form protein. Protein will form the coat, genome will go inside it and new virus babies will move out. But in case of macrophage, the cell is not destroyed, but in case of T helper cell, the cells are destroyed. You can see this line. Infected cell can survive while viruses are being replicated and released. So, what cell they are talking about? They are talking about macrophage. They are talking about macrophage. Got it? Let me explain you one more time. The virus is infecting it, the human cell. RNA enters, RNA form DNA, DNA become double stranded because we have two RNA. Two DNA will be formed, they will join together, form double stranded DNA. DNA incorporates in the genome, DNA undergoes transcription, form RNA. One RNA will become genome, the two strands, right. The other RNAs will form protein coat, new baby viruses will be formed. Okay. Now, I have told you that T helper cell is destroyed. T helper cells are destroyed. Okay. T helper cells are destroyed. So, in a healthy person, the count of T helper cell is 1200 per mm cube of blood. So, if you will check my blood, maybe my T helper cell count will be this much. But if person is HIV infected, then what will happen? The T helper cell count reduces. The T helper cell count reduces. So, we say if a person got HIV today or somewhere, the person is said to be HIV positive. So, simply listen to me very carefully. I am saying if a person have HIV in blood, this virus in blood, you will say this person is as HIV positive. I will not say the person is having AIDS. The person have AIDS, the person will show symptoms when the T helper cell count reduces and T helper cell count in AIDS will be less than 200 per mm cube. If a person is having blood T helper cell count around 1200 or less than that, but more than 200, the person will not show any symptoms of AIDS, the person will not having be any diseases. But if a person shows the symptoms, that means the T helper cell count is little less. Now, what symptoms or what diseases that person got? We call these diseases as opportunistic diseases. So, in AIDS, the person get opportunistic diseases. What are opportunistic diseases? Earlier, these diseases were not happening to you because your T helper cell count was good enough. So, these were very weak, these microorganisms were very weak for your body, like fungus is very weak for the body. So, it was not causing you pneumonia, though you were exposed to it, but your immunity was very strong. But now, you got HIV, you become HIV positive, but later then your T helper cell count start to reduce and it becomes less than 200 per mm cube. So, at that time you start getting opportunistic diseases like fungal pneumonia. Now, we will say you have AIDS, right. So, here during AIDS the person got opportunistic diseases and the T helper cell count is less than 200 per mm cube, right. So, if a person has the better T helper cell count but have HIV, I will not call the person is having AIDS, I will say the person is HIV positive, fine. So, here opportunistic diseases are like fungal pneumonia, tuberculosis, sarcoma, which is a cancer, and toxoplasmosis. Toxoplasma is a protozoan, right, and its disease is toxoplasmosis, fine. So, this is how the person got AIDS, and person never got AIDS on the second day of infection, no. Imagine the person get HIV positive and then person got AIDS. How much time does it take? There is a time lag. There is a time. The time lag is around 6 months to 5 to 10 years. We can also extend the time if you got to know at a early stage that person got infected. How? By giving some immune cells or some therapies. This time can be increased. The person will then not get any AIDS, right? So there is a time interval from getting HIV positive to have AIDS. So it's not like in 1 or 2 days you will get AIDS. It takes time. Minimum 6 months to maximum 5 to 10 years. Fine. So this is how the person is really getting problematic in AIDS. Okay. So how will you detect the person is having AIDS? What are the detection techniques? First technique for detection is Western blotting. In Western blotting, you detect protein. There is a detection of protein. For example, a person is a suspect, that person might get AIDS, right, or might have HIV. Now, how does a person know? Imagine the person had unprotected sexual intercourse with the person, and that person somehow is HIV positive, and you got to know, oh, that person is HIV positive, let's get checked. So that's why you get checked. Okay. So somehow you have a doubt. Now, this is a test that is done, which is a confirmatory test. It's a kind of a confirmatory test. We take the blood sample and we see, is there any GP120, GP41, P17, P24 present in the person's blood or not? If that protein is detected, because that protein is particularly of HIV, the person might have HIV. So it's a kind of a confirmatory test. So we detect the protein. Protein of what? HIV. Second test is ELISA. The full form is enzyme linked immunosorbent assay. This is the assay with A, that means detection technique. Now what do we do in ELISA? Let's see. So what do we do in ELISA? In ELISA we have a plate, and to this plate a protein of virus is attached. It's already attached. This is how you get the plate. For example, GP120 protein has been attached to this plate. Okay, now what we will do? We will take the blood sample from the suspected person. For example, a person named Ram came, and Ram want to get detected. Okay, want to get his HIV test done. So what we'll take the blood sample, because in blood we have HIV, and this is what we are talking about. So we are basically confirming the person is having HIV or not. If a person is having HIV, the body's immune system will get triggered. Yes, because it's not like that the immune system is not working. Immune system is working. B cells are getting activated. They are producing antibodies, but the virus is very strong enough. The immune system is not able to work properly. Why? Right? T helper cells stimulate B cell. B cells are working, but they will not work efficiently because T helper cells are destroyed now. So the person body will definitely produce antibody against the HIV protein GP120. Now we have taken the blood sample. The blood, if a person is HIV positive, we are assuming that person is HIV positive. That person will be having antibodies formed against the viral protein, and that antibody will join to this GP120 protein. So this is a protein of virus which we already have, and this is the antibody from patient's serum. We are thinking that person is HIV positive. Okay, imagine the person is not HIV positive, then this antibody will not be there and it will not join, right? Now we have made, synthetically we have made one antibody, a special antibody that can bind to this antibody, and it also have enzyme conjugated with this. You call it as enzyme conjugated antibody. Enzyme conjugated antibody, but this is synthetically produced. Okay, because no antibody have enzyme with it. Okay, so this is synthetically produced. It has been designed in a way that it will bind to the antibody of a person if a person is having antibody, if a person is HIV positive. Now we will add a substrate to it, and the substrate does not have any color. So when we will add substrate, and if this enzyme is present, substrate will be converted into product and product will show color. Substrate does not have any color, right? Just like sucrose is a substrate, sucrase enzyme, and it gets converted into glucose and fructose. Just like that, substrate will get converted into product in the presence of enzyme and will give purple orange color. So if you will get purple orange color in the sample, the person is HIV positive. If person is HIV negative, this antibody will not be there, this antibody will not join, and hence substrate will not get converted into product. So that's the game here. Person is HIV positive, antibody will be there. If this antibody 1 is there, 2 will join, which have enzyme, substrate will be converted into product. So this is how you can detect whether a person is having antibody in its blood, because if antibody is there, the person will definitely have the virus in its body, right? So this is the second test, which is the ELISA. So this is how you can detect whether a person is positive or not. Now let's talk about the vaccine. Do we have any vaccine or any drugs for HIV? First of all, let's talk about drugs. Yes, we do have drugs. Drugs like reverse transcriptase inhibitors. So we can take some drugs which will inhibit this enzyme, right? Like protease inhibitor. If these enzymes of virus, they are inhibited or destroyed, will virus be able to replicate in your body? No, because these enzymes are very important for viral replication, for attacking of virus to the macrophages. If these are not there, the person or the virus will not be able to multiply in your body, the virus will not be able to grow in your body. So these drugs are given, right? Sometimes alone, sometime in the combination. Then we have the vaccine. Do we have the vaccine? No, we don't have the vaccine. Why? Because virus mutates at a faster rate. At a faster rate. What is mutation? The change in genetic material. So during replication, during every replication, virus will change itself. For example, imagine this virus enters into your body. This have such kind of spike protein, right? It attacked the cells. It attacked the cells, and now this attack cell is producing different kind of a virus. So after every replication, it has changed its coat, and you all know the coat are generally antigen. So first antibody was produced against this virus, because your immune system got to know, oh, that is a virus. Immune system produced antibody against this virus. But after attacking your cell, the new viruses are produced which have different kind of coat. So antibody form will be different. So vaccine is always successful when you have one kind of a virus, so that if the second time virus come, you can kill it. But second time virus is quite different. This antibody will not work for this virus, because it has different coat, different antibody will be formed. It has different coat, different antibody will be formed. So how many vaccines can you give, and how can you predict that what kind of mutation will take place? So that's why vaccine is very, very difficult in this case. Fine. Okay, now let's talk about prevention. So that's a paragraph from NCERT. Usually you guys really ignore this, but you have to read it with me. As AIDS has no cure, prevention is the best option, because as I've told you, there are certain drugs. The drugs can inhibit the replication, but you generally don't get to know you have HIV. You get to know when you get symptoms, and at that time you already have AIDS. So how much drugs can save you? Very few chances. So how can you get cure? You can't get cure, but you can prevent the disease by some awareness. Okay, if you know this is the means by that the HIV enters into the body, you can follow the precautions. Moreover, HIV infection more often spread due to conscious behavior patterns and is not something that happens inadvertently like pneumonia or typhoid, right? So you know, usually this happens due to your behavior that you're doing, like unprotected sexual intercourse. It's not like you're eating food and you have to eat food and somehow it's infected and you got it. Or some person is having pneumonia, you get the pneumonia, right? If you know it is spreading through sexual intercourse, why not to just use condoms? And one another thing, do not go for multiple partners, because you never know. If you are changing partners every time and those partners are also unknown, you are more likely to have AIDS, right? Of course, infection in blood transfusion patients, newborn, etc. may take place due to poor monitoring. Now, why the poor monitoring is a case here? Imagine the doctor is giving you blood. The doctor is not checking whether it is HIV positive blood or not. That means it's a negligence of a doctor, right? Or we can say newborn babies got it. If first of all, in India or in any country, when a female is pregnant, she has to undergo a lot of tests done. One of a test that is done during first trimester is HIV test. So they confirm whether a female is having HIV or not. Accordingly, the pregnancy will proceed, right? Otherwise, the baby will also get it. So these are due to, if for example the doctor didn't tell you, so that's a kind of a poor monitoring. The doctor didn't ask you to have test, that means it's negligence. The only excuse may be ignorance, and it has been rightly said, do not die of ignorance. So many of people die in AIDS due to ignorance, right? You don't know that you have, or whatever act you are doing, you are not at false side in that. For example, if a doctor is giving you or a nurse is giving you wrong blood or HIV positive blood, you are not at all at mistake. But if you ask the doctor, have you done the HIV test on the blood, then that means you can save yourself. So do not die of ignorance is a slogan for AIDS. In our country, the National AIDS Control Organization NACO and other non-government organization NGOs are doing a lot to educate people about AIDS. So one organization is NACO, and other NGOs are also working for spreading awareness. Many people even does not know about AIDS. So you have to teach them. Yes, this is how it happens. So these type of organizations, they also go to brothels and all, distribute condoms, aware people around that how this disease is deadly and how you can protect yourself from that. Nowadays people also get awareness through advertisements and so, right? WHO has started a number of programs to prevent the spreading of HIV infection, making blood safe from HIV, ensuring the use of only disposable needles and syringes in public and private hospitals and clinic, free distribution of condoms, controlling drug abuse, advocating safe sex, and promoting regular checkups for HIV in susceptible population are some such steps taken. So WHO has made some mandatory steps in the countries, like if there is a blood in the blood bank, you have to get it tested. You have to get it tested for HIV, right? Second thing, there should be use of disposable needle. Whenever you go to hospital, the doctor will always open new needle from the packet and then the doctor will use. That's a kind of a norm that is given by WHO, that you cannot use the needles again and again. For every patient you have to use the new needle. So for that, plastic disposable needles are generally used. And syringes in public and private hospital and clinics. Free distribution of condoms. So in many areas I have told you, like NGOs distribute free condoms. Controlling drug abuse. If you will stop people from using the drug, as I have told you, they use needles, right? They share needles. That also lead to the spread of disease. First of all, never take drugs, because with that you have a gift also, right? Second, advocating safe sex, telling people that if the person is unknown, never ever go for unprotected sexual intercourse. First you can get AIDS, second you can give baby, right? And promoting regular checkups. So for example, there is a population and there are a lot of people who are HIV positive there. So to that population, it's mandatory that they will undergo regular checkups for HIV, right? Now, infection with HIV or having AIDS is something that should not be hidden, since then the infection may spread to many more people. If one person is having HIV and that person is also undergoing sexual intercourse with the other, you should definitely tell that person. Many people don't tell it because it's a taboo. They think what they will think about them, but you can take another person's life. So never keep these things hidden, you should be open to it. Alright. HIV AIDS infected people need help and sympathy instead of being shunned by the society. And also, if they will tell, there are people who have a mentality, oh that person is HIV positive, don't sit near that person. You will never get the HIV by just sitting next to that person. You will never get it by the sharing of food. Neither you will get it by saliva. You will also not get it through the mosquito and so, right? These are not the ways. There are certain ways by which the virus move inside body. Unless society recognizes it as a problem to be dealt with the collective manner, the chances of wider spread of the disease increase manifold. So, if you will keep on doing that, then people will not tell and the disease will spread more. It is malady that can only be tackled by the society and medical fraternity acting together to prevent the spread of a disease. So, people and doctors and all the health workers, they have to come together to stop this disease. Any of these, if they don't work harder for that, the disease will keep on spreading, right? So, that's about AIDS, guys. That's about AIDS. You know when is AIDS Day? AIDS Day is celebrated 1st December. So, when we were in our colleges, we used to celebrate AIDS Day by the candle march. I remember, I have some pictures of that also. I will not share, obviously not here. So, I remember, I have a memory of that when we were in college. We used to go into the entire sectors, so in the Chandigarh, right, and entire sectors and spread awareness. We had a banner and so. So, this is how you can spread awareness. You can tell other people who are not educated and you can spread the awareness. It's always good, if you have a knowledge, always spread. Anyways, let's solve some questions. Find the incorrect statement with respect to AIDS. HIV is an enveloped retrovirus. A widely used diagnostic test for AIDS is enzyme-linked immunosorbent assay, ELISA. Third, in AIDS, the count of B cell reduces. Fourth, AIDS can be spread by unprotected sexual intercourse. We have to find the incorrect. First says HIV is an enveloped retrovirus. Very true. If you have seen, the HIV is enveloped, it has a coating. Why it's a retrovirus? First of all, the genome is RNA. And from RNA forms the DNA by the technique known as reverse transcription, and the enzyme is reverse transcriptase. So, this is absolutely correct. A widely used diagnostic test for AIDS is ELISA. This is also true. We have just done it, the ELISA, yes. In AIDS, the count of B cell reduces. T helper cell. So, this is incorrect. This is what we have to find. AIDS can spread by unprotected sexual intercourse. Yes, but with the infected person. So, partly it's also right. So, answer is 3. Incorrect is 3. Next. Alright. At which stage of HIV infection does one usually show symptoms of AIDS? So, it's the PYQ. First, when the viral DNA is produced by reverse transcriptase. Second, within 15 days of sexual contact with an infected person. When the infected retrovirus enters host cell. Fourth, when HIV damages large number of helper T lymphocyte. Okay, so it is saying at which stage of HIV does one usually show symptoms of AIDS? That means a person start getting diseases. So, whenever the DNA replicate, that means the person got the infection today, the DNA the viral DNA will start replicating today. So, this is how you don't get to know that you have AIDS. You might say you are HIV positive at this point because you have HIV in it, but you can't say you have AIDS. When within 15 days, no, the time lag itself is 6 months to 5 to 10 years. So, this is not the case. When the infected retrovirus enter host cell, imagine you got infection today, maybe today the virus start getting into the host cell, then you don't have AIDS, but you will get starting having symptoms when the T helper cell count is less than 200 per mm cube. Usually it is supposed to be 1200, but here it is 200. Okay, so the fourth is the most appropriate answer. Next. HIV that cause AIDS first start destroying. No, simple question, which cells are destroyed? T helper cell. Answer is 2. Not even macrophages. Macrophages are factories. They are producing there. The cells are not being destroyed there. Oh, okay. So, the macrophages are HIV factories, no cell destruction. Thus, whenever you got the term destroy, Simply mark T helper cell. Which of the following is correct regarding AIDS causative agent HIV? It is undeveloped retrovirus. Second, HIV does not escape but attacks the acquired immune responses. HIV is enveloped virus containing one molecule of single-stranded RNA and one molecule of reverse transcriptase. HIV is enveloped virus that contain two identical molecule of single-stranded RNA and two molecule of reverse transcriptase. So, we will start with the first. You have to find the correct one. Is it undeveloped? No, it is developed. It is causing AIDS. So, how can you say it is undeveloped? It is highly developed. So, this is wrong. It does not escape but attack the acquired immune response. The virus, if it escapes, only then it can attack the immune response. If it is not escaping, that means immunity is higher at that point. Escaping means to get away. To get away, because it is getting away, that's why it is causing you disease. So, this does not is wrong. It escaped, that means it is moving away from immune system. Immune system is not able to recognize it nicely, right? So, that's why it is causing you disease. So, it is wrong. HIV is enveloped, true, but it does not contain one, rather two identical molecule of the RNA as well as enzyme. So, answer is 4. How will you get to know there are two molecule of reverse transcriptase? Okay, let us see here. So, in both the molecule I have drawn two enzymes. Okay, alright. Let us move to the next. So, that is it about AIDS. So, listen to me very carefully, students. You might be thinking, no, it is just a disease, they are not going to ask the question, but be ready. You will get the question from either AIDS or cancer in your exam. Cancer will be doing in the next class, AIDS we have done. So, please read NCERT very carefully and read each and every line of this, because it is quite important, right? And even the diagram, that is why I brought the diagram from NCERT, because sometimes they give you the question out from the diagram, then what will you do? You have to solve the question, there is no choice, fine. So, read the NCERT, learn the AIDS very nicely, because one question from AIDS, one question from cancer. You already know this is one small topic and you will get the question. Why not to do it nicely? Okay. So, I will meet in the next class. We will study cancer. Till then, bye-bye. Take care and study hard.